introduce the last talk of the day and second keynote speaker, Dr. Georgiana May, who's a professor at the University of Minnesota in Ecology and Evolution, as well as in plant biology.
It's on.
A little bit of background. Dr. May received her undergraduate degree at Bowling Green, actually, and then a master's in the University of Georgia with Ruth Haynes. And then she was then John Taylor's first PhD student, where she worked on concepts of individuality in mushroom fungi. And then she did a postdoc at UNC, where she was the first person to clone mating type genes from mushrooms. And then in 1991, she moved to the University of Minnesota, where she's stayed since. And so I've known her for quite a while, and she certainly has been a great inspiration as I was doing my own thesis on mating type genes. And she still has published the most important work in that, where she estimated the strengths of balancing selection using intra-allele and within allele differences. But let me just cut to the chase. She's done so many different things, and now she's doing really interesting things on endophytes. And it wouldn't be a proper symposium if we didn't actually consider endophytes, because essentially this is sort of the plant version of the microbiome that we've been talking about today. And so without any further ado, please join me in welcoming Dr. May.
Well, thank you for that very generous welcome. You know, when Tim invited me to speak, and I heard that it was an early career scientist symposium, I said, well, I could do that, because I've only been in Minnesota over 20 years, but I'm still early. And then I read the program, and I found out I was supposed to be a keynote speaker, and I thought, oh my gosh, I'm supposed to impart some wisdom here. So I thought I could do that, but I had to dye my hair gray first, so you believe me. So I'm trying to fill both shoes, and I have to say it's just been a wonderful symposium, and especially the energy that the young presenters bring today is just fantastic, and I feel quite honored to be in your company and really enjoying the presentations. I also realized I'm the last person today, so my presentation does have some detail, but unlike the early career scientists, I don't feel compelled to give you every methodological detail, and I'll be happy to answer questions at the end. So my general question here is whether microbial interactions within hosts might drive the evolution of increased virulence in a pathogen. And so I started working on this problem because it was a conceptual issue that confronted me as soon as I realized the extent to which all host organisms are occupied by so many different multiple organisms. So as you all, or another way of looking at this is how do these multiple organismal interactions affect pathogen fitness? So as you all know, that the organisms are full of these microbes, and we call this the microbiome. And this includes animal systems, and I'm glad there's another plant representative here. Plants are also full of their own microbiomes. And you know, this has actually been portrayed in our artwork for a long time. This is a picture of Eden. What I like about it best is these domesticated animals here that are pictured. So presumably, ever since the creation or the evolution of these organisms, they've had microbes, which have actually a lot of these do move amongst their hosts. So the microbes and their hosts live in these diverse communities. And therefore, they have these very diverse interactions. So just as an example of that, the system that needs no introduction here, something like Wolbachia, an intercellular symbiont of many insects, actually occupies a lot of different kinds of insects with a lot of different kinds of effects, especially on their reproduction. So many of these would be considered parasitic effects. But we can also see that over long periods of time, there's probably been quite a diversity of host interactions that have evolved. So in flies, the Wolbachia might provide defense against a parasite, spiroplasma. In flies also, Wolbachia tend to decrease in virulence and increase the host fecundity. Today, mosquitoes Wolbachia is being used for protection in mosquitoes against the dengue virus, trying to decrease the transmission of that virus to human hosts. And so, and there's also some theory emerging now that, such as this by Fenton, which would suppose that if the symbiont can provide some benefits that outweigh the costs, because most of these symbionts actually have a cost, then it can be stably inherited and evolve with its host. So to me, there's two really big things that we need to think about in this field. One is this long-standing theory that would suggest that co-infection, that is by different species, or multiple infection by the different genotypes of the same species, should select for greater virulence. And that's mostly thought in terms of the pathogens competing with each other for host resources. And a virulence is a mechanism of getting those host resources first or fastest. So that leads to this paradox, though, that I think we're only really beginning to recognize, which is we are, in fact, filled with these microbes, most of which do not have a detrimental effect on us. So they can't all be driven to higher virulence, or we'd all be dead and not here talking today. So I think part of the answer of that is that, or at least part of the answer to that is that these environments in which these microbes live, actually the ecological context varies quite a bit in space and time. And we know also from ecological theory that that should limit the evolution of virulence and maintain diversity in these systems. So I've structured my talk really around thinking about fitness effects and about ecological effects. Just to develop those a little bit more, that trade-off between transmission and reproduction, so this is the classic May models, should produce an intermediate or optimal virulence, and then competition amongst multiple infections should select for increased virulence. So Ebert and Bohl wrote this review paper in Trends in Microbiology that had some nice figures. And what you should get is a optimal virulence at which you are, you're optimal for the parasite. You get the most reproductive fitness with the least amount of damage to the host. In this case, virulence is measured as parasite-induced host mortality. And then under multiple or co-infection, that should shift so you should get this evolution of increased virulence. So in this talk, and the work I do in my lab, we want to ask whether these symbiont interactions affect that evolution of virulence toward a shared host. Okay, so then there's this ecological attribute that should affect the evolution of virulence. And so we know that these microbial species vary across even an individual, as many people have illustrated today. They vary within an individual in different locations. They vary among individuals within a location, within a spatial location, and they vary amongst individuals across locations. So in this case, these people, and actually as it turns out their dogs, share a lot more of their microbiome than any group of people across a city. But even a city is structured, and so there's transmission of this microbial species amongst these people, and so the structure of the microbial communities then depends on this transmission amongst hosts. Similarly for plants, we can show tremendous variation within and across individuals, among individuals within a location, and among individuals across locations. So I purposely have used May's for this example because that's the system that I'm that I'm working in and that I'm reporting on today. But, you know, you can also have other examples of these microbes varying. And I think this is the first example of a plant microbiome. So you have your gut fungi here, your oral microbiome here. And undoubtedly, these microbes are varying across these hosts. All right, so again, in this structure, this ecological structure that might both limit the evolution of virulence and maintain diversity, we have a lot of support for this idea in the theoretical literature. For example, Ben Kerr's work with the emergence of a more rapacious mutant. For a while, it comes up to a greater frequency, and then it dies out. And so it has this sort of dynamic, and in similar experiments he did in microtiter plates, actually. Without any structure, then the vicious phage takes over. But if you have restricted migration, these two organisms can co-evolve, and both are maintained in these populations. That's on a microbial scale or a very small spatial scale. But we can also see these effects in large macroorganisms. For example, rice planted to different varieties in China had a lower incidence of rice blast than monocultures of those plants. So we want to ask how the ecological context of these microbial interactions might affect the evolution of virulence in the pathogen. So today what I'll go over is some of what we know about the ecology of these microbes. I guess a disclaimer is that I'm not actually an ecologist. And then I want to talk about the experiments we've done to look at the effects on fitness of the pathogen of these interacting organisms, and work we've done to identify traits governing these microbial interactions, and asking whether these also are affecting pathogen fitness outcomes and symbiont fitness outcomes. And then lastly, try to put this back into this ecological context. So this work I'm reporting here started with a postdoc in my lab, Jean Pan. And so I'm also trying to sell you all to plant systems, because plant systems really provide these replicable experimental systems, right? And you don't have to have permission to do all kinds of things to your plants, and when you're done, you put them in a compost heap, or when you're done with your microbes, you autoclave them, and you're good, right? No permits really are needed. And so in this case, what I'm talking about today are these fungal endophytes. And actually all the microbes I'm talking about today are fungi. So we define endophytes sort of pragmatically. That is, we take healthy plant tissues and we isolate microbes, and those plant tissues are not showing any signs of disease. That classifies it as an endophyte if it's living internally to the plant. We also, for all the experiments I'll talk about, we've taken these microbes and put them back in the plant on their own and shown that they don't cause disease symptoms. So they are at least commensal organisms. And then my pathogen of choice is Eustalogomatus, the delightful blight. This causes these huge hypertrophies on any part of the plant. It's shown here on the ear. These galls, as I'll call them, are filled with spores, black spores. And so we can use this because this is all of the reproductive effort of the pathogen, we can just collect and dry and weigh these galls and use that as a measure of reproductive fitness. So again, we have this microbiome that does differ within and across individuals, among individuals, and across locations. And we've done we've done some work to show that, but what I really want to focus in on today is these ecological contexts that may affect the interaction of these microbes and then the evolution of virulence. So I'm going to, partly because I'm trained as a mycologist, and mycologists just love life cycles. You have to have a life cycle in your diagram. I mean, in your talk, ever since I was a graduate student, okay? But I also like life cycles. So Eustalogo has to mate to infect the plant. Here's the plant. in this drawing. And then it does make a structure that allows it to penetrate the surface of the plant, and then it ramifies through the inside tissue of the leaf or the plant tissue. And it grows, and then finally it induces this hypertrophy of plant tissue called tumor or gall, which is producing these black spores. The black spores undergo meiosis, and the cycle is repeated. What I'm using this for today throughout this talk is not just so that all of you can become amateur mycologists, but because Eustelago interacts with fungi or other microbes in the plant, it interacts with microbes on the plant, and it interacts with microbes in the soil. So this is this ecological context that I want to emphasize here today, is that these are three very different environments, and they actually have very different fitness outcomes for these organisms. So first of all, do we even have evidence that the interactions amongst these microbes could structure these communities? So that's so I'll show you some data that suggests that these microbiome species may either facilitate or limit pathogen growth within the plant and the distribution of of the pathogen across plants. So these samples were taken at 2 locations in Minnesota, one under continuous field corn, which is resistant to Eustelago matus, and one on a continuous cultivation of an inbred that's susceptible to Eustelago matus. So when I talk about susceptible and resistant hosts, I'm talking in terms of the relationship to the pathogen. So in these fields at 20, even though this is resistant field corn, you can find occasional infections. And we sampled adjacent plants that either had galls or didn't have galls, and we sampled each of these in the same way. So we can get both a fine scale and a large scale distribution and co-distribution of these organisms. And we did a lot of culturing, and we've also done non-cultured sequence-based OTU calling, okay? But I like to look at all the pretty colors. So the test that I'm just going to show today is one called a checkerboard analysis. So basically, if these organisms are, if you have Eustelago present, is a second species absent, or if Eustelago is absent, is the second species present. And we're going to compare a pattern, a checkerboard pattern like that, to that which would occur by chance. And if we see a difference, that is that they co-occur less often than expected, you might infer antagonistic interactions between them. In contrast, if Eustelago or the species 2 are facilitating each other's presence in the plant, they should co-occur more often than expected by chance. The inference of antagonism or facilitative interactions is an inference, and we've actually checked a lot of these and found that they do match these general patterns. But here I just want to talk about the general patterns we observed. So in that resistant host, in fact, we found that whether you take commonly occurring endophytes, so these are a wide diversity of taxa, whether you look at fusarium species, species by species, fusarium's the most common endophyte in these populations, or whether you group them by the metabolite they produce, they are co-occurring with Eustelago more often than you'd expect by chance, and use a randomization test for that. So it suggests that these things are, at least are not antagonizing each other's present in the plant when they're present in a resistant host. We found almost the opposite result in the susceptible host, where, although the species by species for fusarium, it wasn't significant. The most common endophytes, and if we group the fusarium by metabolite, seem to more often be antagonistic to each other's presence in the plant. Okay? So this was interesting, and actually not exactly expected, except that if these things are co-evolving in these fields, then both Eustelago and these endophytes are getting into this susceptible host more often and competing for that host resources. So so what I want to build in this talk is a sort of an alternative view of the evolution of virulence, which is, if we think only in terms of the host, for which most models are developed, We would expect that a resistant host will select for higher virulence so the pathogen gets in, or so that the pathogen ramifies in the host. And if a susceptible host should select for lower virulence because otherwise the host is killed and neither organism reproduces. I think when we start including the microbial symbionts, we have to think that in a susceptible host, the pathogen and symbiont are meeting more often. The pathogen is able to get in. The symbiont distribution is about the same for these two kinds of hosts. And so this competition should select for actually greater virulence. That's true if virulence and competitive traits are correlated. That's something Ebert and Boll brought out in that original paper. And so we have some evidence that that's true, but it doesn't quite play out the way you expect. So then if we're going to predict the direction of selection and any evolutionary changes, we have to have some real honest-to-gosh measure of fitness. So in this set of experiments, which is pretty extensive, what I've done is used what I call model microbiome, which I'll explain to you. It's very simplified. And we want to ask whether the competition between a symbiont and a pathogen actually causes this or engenders this selection for virulence, greater virulence. And what we find is, so giving you the take-home message first, is that these fitness effects due to the symbiont are measurable, and the direction of those fitness effects, the fitness outcomes, and the direction of the selection vary over the plant, across the plant and over time, depending on these microbial interactions. So let me explain that. So here's our model microbiome, is the host plant corn, which you're probably all familiar with, Eustylogomatus, which some of you may not be familiar with, but it's a very nice thing, don't you think? And then my model Endophyte is Fusarium verticilloides, which is the most common fungus in maize. And what we have here is the cell walls of the plant stained red, and this is how the Fusarium grows through that plant. So this is and most of the time now for the rest of the talk, I'll just talk about an endophyte and as this organism. Both of these fungi are what we call biotrophs. They require a living host for reproduction and growth. All right, so we can set up some various thoughts or hypotheses for this. If, oops, wrong button. Say if these two organisms are competing very strongly for host resources, then we might expect that to select for greater virulence, with virulence as a mechanism to gain these host resources. But if the symbiont in any way can reduce the cost of that virulence, then you could select for lower levels of virulence, okay? And then we, on this sliding scale here, which all organisms occur along, you'll notice that our symbiont actually becomes what we call a defensive mutualist, right? So this first set of experiments I want to explain were done by Kun-Sub Lee, a graduate student in my lab at the time, and some additional work I'll explain that Alma Rodriguez Estrada, who is a postdoc in my lab. And in this case, what we're doing to look at that relationship between these interactions in pathogen fitness, what we did was generate 24 Eustylogomatus strains of varying virulence. And again, fungi are great for working on because these are all clones of each other, so we can, and basically we can get plants that are basically clones of each other, so we can control the genetic variation in this interaction. And then Kun-Sub took two different Fusarium genotypes that at the time he thought were pretty different from each other. They turned out not to be, but I'll report the data. And then we gather up, as I said, we gather up these galls and dry them as a measure of Eustylogo fitness. Okay, just to convince you that they did vary in virulence and to define how what I'm calling virulence, I'm calling it as a reduction in plant fitness, so it's more of an animal animal system definition of virulence, which I think is appropriate. In this case, we're using plant size as a proxy for that. So the control plant here doesn't have any fungus in it at all. And they grow to about 60 centimeters tall. And then if you focus on the black bars, that's plants that are inoculated with these differing Eustologo genotypes along here. Okay, so if you look from the least virulent, it actually didn't cause a significant decline in plant growth to the most virulent, where you have plants that are half the size of a control plant. Okay, so this is a significant effect. These are pathogens. They do damage host growth. The purple bars, we don't need to go into too much detail here, but you'll notice that in general, plant height is greater so they represent plants that were co-inoculated with the pathogen and the symbionts. And you'll notice in the greater part, they're growing higher than they would if they're inoculated with the pathogen alone. And so we're defining this decline in plant growth here as virulence, and that's how I'll use it for the rest of the talk. So here's some data, and boy, did I love seeing all that scattered data people have. So when you start really talking about real organisms, this is what happens. And you know, it's actually, when I first saw these data, I was a little bit disappointed. I'd say, where's my trade-off? You know, where's my, you know, you can't see it here, but what was interesting is that I gradually realized that we used a score for life history stage, and these data are breaking out by life history stage of the pathogen as it develops in the plant. And so these here, where there's no reproduction at all, so all these graphs you'll see, most of them have pathogen fitness as the log of the total gall weight produced on an individual plant. Each plant is a dot, so we had a lot of replication. And then here's our virulence of each of those individual genotypes, so that's why these are in columns. So we have replicate plant inoculations with the same genotype. So a large, some of these fungi never get in the plant, okay, so there's no infection or growth, and therefore no galls. So that's the first stage of development. The pathogen has to get in the plant. These orange dots are because there's slower infection and growth, and the galls are fairly small, and these are often on leaves. The majority of the data are up here in the blue and the black, where both fungi have gotten into the plant and the pathogen is reproducing. So I'm going to use this diagram as showing you what all the data, these are real data, okay? So I'm going to use this throughout the talk and then break down the fitness effects by these life history stages, infection, growth, and development of these galls. So I'm going to do that, and so this is the same data. We have the virulence of each Eustologo genotype, and we have pathogen fitness as the log of the total gall weight per plant, and each dot is a plant. But now I've recoded it, or recolored it to show the experimental variables. So the black dots represent when there's a pathogen of the Eustologos inoculated, but neither of the endophytes. And the two purple dots are the pathogen with one or the other endophyte. And the endophytes actually make these colors in culture, so that's why I use those colors. All right, so now I'm going to walk through the fitness effects of this life history development and the interactions of these microbes. So first of all, infection. So this is the estimated or the predicted infection rate. These are the real data. So if it's 0, there's no infection, and if it's 1, then that individual plant was scored as being infected. We can look for the symptoms of infection. There are many dots here represented, so this is greatly overplotted. But the prediction is that if you look at, here's Eustologo, the pathogen infected just by itself, and you know, even at a low virulence genotype, the plant gets infected, some about 25% of the time, and then most of the plants become infected. But the effect of the endophyte is really interesting. It's suppressing infection. So remember, this happens on the surface of the leaf. or on the surface of the plant. So for both of these endophytes, when they're co-inoculated with the pathogen, they significantly depress the level of infection. And so if you can see that if you look for 50% infection, this requires almost a doubling of what we estimate is the effect on the plant growth of virulence, okay, to get the same level of infection. So in this case, the presence of the endophyte should actually select for greater virulence. It should drive the virulence to higher levels. This single effect at this early stage of pathogen development. That big middle ground here of slow developing pathogens is now taking all that data out, and that's just this data here. We coded it as a life history stage 2, and now it's colored by these pathogens with and without endophytes. And so there's a huge amount of scatter here, which actually, at first, I think when you see a lot of scatter in your data, you're kind of disappointed. But I find this interesting. I think noise is data. So what this is telling us is that in these microbial interactions, there's actually a lot of stochasticity. Maybe one of them got to the leaf or the surface of the plant a little bit earlier than the other one and established its turf, so to speak. And so there is going to be this tremendous variation. So there's hardly any relationship. This is a very almost a flat slope, so very little relationship to virulence. But these intercepts are significantly different. So even with all this scatter, so the pathogen alone has a higher level of reproductive fitness at this life history stage than if the pathogen is in the plant with these endophytes, okay? So now at this next stage of development, the endophyte is still suppressing the development of that, overall, still suppressing the development of that pathogen. And if we did some qPCR analyses, so we're using qPCR as an estimate of biomass in the plant. And so if we look at growth of either of these fungi alone in the plant, we use that as our standard. Now we're going to compare to what happens when the two organisms are together. And so not surprisingly, the pathogen is suffering quite a loss in ability to grow in the plant. It's growing much more slowly than it would if the endophyte wasn't there. But what's more surprising is that the endophyte actually gains a growth benefit from being in the plant with the pathogen. So the endophyte grows better in the plant with the pathogen than it would alone. And this is quite a large increase, okay? We actually see this not only in the plant, but in saprophytic interactions in a petri dish. So the endophyte is getting a benefit from whatever it's doing in the plant. All right, and so then finally to look at this last class of development. So now these are well-developed galls. The pathogen and the endophytes have gotten in the plant, and it's the largest part of the data. Most of the data points are here. If we look at that, even with all this scatter, we get a significant difference between the effect of the pathogen by itself, the black line, and a simple linear regression through these points, and the pathogen in the plant present with the endophyte. All right? Those, I'll explain in a minute, those look like small difference, but remember this is a log scale. They actually represent about an 11% increase in pathogen reproduction in the presence of the endophyte. So you should be scratching your head about now, because I just told you that the pathogen actually slows its growth in the plant when it's there with the endophyte. So in fact, this is a sort of counterintuitive result. I should point out the largest, most interesting thing is that there's a decline in fitness with increasing virulence, so as we'd expect by a trade-off. So this difference is not only significant, but it's you know, a sizable chunk of reproduction, about 11%. But how do we explain that result where you've actually increased the reproduction in the presence of the endophyte? These galls are producing the most spores. So as we've just seen, the endophyte does slow the growth of the pathogen in the plant. Because of that, the plant lives longer and grows larger. And pathogen reproduction directly correlates with the size of the plant. So even though the endophyte is actually slowing the growth of the pathogen ends up benefiting in terms of reproduction. So if we look at these two effects together, we have this slowing of infection or a decrease in infection rates, which is occurring on the surface of the plant. That should select for increased virulence. But not only do we have this trade-off of fitness and virulence, which will increase, it will select for a decreased virulence, but we'll get a growth benefit or a reproductive benefit from being in the plant with a endophyte. And so those two things together should select for decreased virulence, because basically the endophyte has ameliorated some of the effects of the pathogen on the plant. So that optimal virulence then will shift in response to the microbiome species that this pathogen encounters as it lives in the plant. And the optimal virulence then depends on ecological context. Overall, if you have a very varied spatial distribution of these endophytes, which we know we do, then you should expect a slowing of evolution of greater virulence. And then going back to this on a very small spatial scale, the endophyte interferes with pathogen infection, and because once they both get in the plant, both the endophyte and the eustallago, or the pathogen, gain, there's actually sort of a conflict of interest here. And I'm going to, toward the end of the talk, I'll try to explain why I think this gets set up, because I think it's interesting. But right now, what I want you to see is that these fitness outcomes are very different over a very short distance, the outside and the inside of a plant. And they also differ over that life history of the pathogen. So these things are varying in their interactions over very small spatial scales and over time. All right. So, you know, that's actually, I feel pretty proud of those measurements, because they were difficult. These experiments are actually pretty difficult to do. We had whole greenhouses full of plants, and you have to get all your organisms growing properly and all this stuff. So I was happy with those results. They are broad brush. They're telling us fitness outcomes, but they're not telling us underlying processes or mechanisms of those outcomes, like why do we get these results. And so what I wanted to do next was look at traits that might govern either these different fitness outcomes, either facilitative or competitive interactions. And I want traits that are both affecting these interactions and affecting these fitness outcomes. And so what I wanted to do then was to compare these organisms just growing together in saprophytic growth, which is a petri dish, with how they're interacting in a plant, okay? And Alma was kind enough to join me in this adventure. And I also was collaborating with Corby Kistler and Wilfried Yonkers at University of Minnesota. So again, we're looking at the interactions in saprophytic conditions called a petri dish. Here's Fusarium interacting with eustallago. Here's Fusarium growing by itself. Here's eustallago growing by itself. And so we were comparing these interactions, or the lack of them. And we were doing both gene expression using RNA-seq. Actually, we did this first set of experiments with chips, if you can believe it. Alma did metabolic profiling, which turned out to be very informative, and we were doing these qPCRs for biomass. We also, in the plant system then, we were co-inoculating or inoculating individually Eustologo and Endophytes. We're using a much more restricted set of genotypes because of the analytical processes we're doing here. We did qPCR for both of these fungi, and then for the rest of these, for the metabolite and RNA-seq analyses, we chose samples for which both organisms were represented in the plant at about the same titer. So just to go through those now, what I'm doing is synthesizing a lot of this metabolite and gene expression data, and I'm not giving it all to you because it's, of course, a lot. So we have, when the two organisms are just interacting, the pathogen and the endophyte in a petri dish, one of the first things we discovered, which was kind of surprising, was that Eustologo is upregulating these small secreted proteins, which are thought to be signals with a host in response to the presence of the endophyte. The endophyte is upregulating attack. It makes these toxins, and it also makes cell wall degrading enzymes that affect Eustologo, and then it's protecting itself with a class of proteins called lyse-M that basically protects its own cell wall from detection. It's also producing this fusaric acid, which we've noticed throughout our experiments is important. Eustologo is trying to defend itself by upregulating transporters to pump whatever. We don't know what, but it's upregulating transporters. It's losing its protective shields, though, because these extracellular matrices are downregulated, okay? And it's also got its what I call its emergency responses up. It's in trouble. So these iron siderophores are thought to be protective against reactive oxygen species, heat shock proteins, and hydrophobins on the cell wall. So it's upregulating its defenses, it's, for the most part, and used fusariums on the attack. When we put then this whole system into the plant, what I've got here is what we just saw, and I'm going to show you how it's different. First of all, we don't see all of those sort of emergency responses, but we also don't see all the attack mechanisms of fusarium. It's not making those toxins. Eustologo has got some of its transporters upregulated again. And it's also generating these small secreted proteins, which classically have been thought to involve interaction with the plant. And these are well described in the literature, okay? So the only interesting thing, or one interesting thing to note here is this square, which just represents one particular effector, which seems to both be signaling in response to the plant and in response to fusarium. It's making its own cell wall degrading enzymes against the plant, and then here's the plant, of course, is responding. And so the plant upregulates a protein that resists these particular cell wall degrading enzymes, and then upregulates a classic NB-LRR resistance locus that is thought to be responding to an effector. We don't know which one, but it is, so these are classic resistance factors that trigger plant immune responses. So here was the surprise, another surprise in this. Not only is Eustologo signaling its enemy as well as its food, but its enemy is causing an upregulation of the same plant immune factor, okay? And so that's totally unexpected. For one thing, these are thought to be very highly specific. And another thing that's happening here is that both of these participant microbes are inducing a greater resistance response in the plant. So I had to break this out. What I've shown you here is sort of more of the physiology. So we know that Eustologo matus uses sucrose directly from the plant. And we also know that it induces dimboa, which is a plant defense compound, which breaks down to spontaneously to some other boa compounds which inhibit Eustologo's growth. Okay, so here's another surprising thing about these interactions. Fusarium has still got its attack and defense things going, apparently in response to use of the logo, but that it has two clusters which encode for enzymes that actually break down the plant defense compounds. All right, so it's this endophyte is actually modulating plant responses and plant defenses in two different ways. The R genes that I just showed you here and in plant defense compounds. All right, so summarizing, what is pretty complex data here, we had this model and we suggested that if we have a lot of interference competition, we should push greater virulence, and if we could reduce the costs of virulence, then we'll tend to reduce the evolution of virulence in the pathogen. So both of these things are occurring in the system, so you should come to a more intermediate and a lower virulence than you would without the interaction of these microbial symbionts. If we then look at the traits that are involved in this, of course we have the classic plant resistance and the pathogen virulence, and there's nothing different about these organisms than you think of in host-parasite interactions. What's really interesting are these microbial symbionts. And so this endophyte is both modulating plant resistance, and I showed you in two different ways, and it's also modulating these costs of virulence by slowing down the growth of this pathogen in the plant, okay, and the plant grows to a larger size. So we have sort of a tug-of-war going here, and instead of these very deterministic models for the evolution of virulence in hosts and pathogens, I think we have to start thinking that really this interaction, because the endophyte is in such a central location or position in this interaction, is what might be the most important determinants, some of the most important determinants for the evolution of virulence. And then on this more local scale, we have sort of a what seems to be kind of a conundrum. So we have this endophyte that's actually not acting in its own best interest, right? Because it gains a growth benefit from having the pathogen in the plant. And we showed that previously, right? And the pathogen actually gets a benefit, reproductive benefit from being in the plant with endophyte. So, but yet we have this interference here at the surface of the plant. And again, these fitness outcomes are very different over a very small distance. So, for a long time I thought, what the heck? Why doesn't, we do have strains of Fusarium that seem to facilitate entry of Eustalago into resistant plants. All these experiments were done in susceptible plants. So, if you're a good organism, why don't you facilitate your food source, which apparently is Eustalago? So here's what I think is happening, is that both these organisms occur in the soil. Okay, so now we've got a really different environment, right? And I've isolated them from soil, so I know they're there. Okay, and so is there a selection for interference competition in the soil that's actually what is going on here on the surface of the plant? I didn't really have much evidence for that for a long time until I started looking at this. And so we have strains of Fusarium that differ in the amount of fusaric acid they produce. That's what's shown here. And then we've co-inoculated with 5 different Eustalago strains, 5 or 6. And what you can see is that the percent of plants affected goes down with the increased production of fusaric acid in these different Fusarium strains. All right? So what I think is going on is that this selection for interference competition in other environments is interfering with the interaction of these, or the selection on interactions on the plant surface. So the Fusarium actually is experiencing very conflicting selection pressures in these 2 very different environments is the way I interpret these data. So this direction of selection then depends on ecological context. So where I'm planning to go from here is to further quantify these conflicting selection pressures and use modeling to demonstrate that they should, as we expect intuitively, limit the evolution of virulence. I'm especially interested, even though I've used a single model microbiome system, is in the complexity and diversity of these microbiome systems. And what I'm thinking is that the rate of virulence evolution should be inversely related to the complexity of those microbiomes, and I think we can study that empirically and experimentally. And then, so then we're going to still be left, or we're still left with this question of why do we even see disease if these microbiomes are so good at protecting us against disease? And I'd like to look back at a more ecological model that occasionally these pathogens just escape their competitors. All right, so where I want to go in some of this work is that I'm getting into natural systems and have two new projects in natural grassland systems. And then as part of those experiments, we're doing what I call microbiome addition experiments to test this complexity effect, and we're doing some modeling. So thank you for listening, and I'm thanking my lab members, Paul Nelson, who's doing some modeling, Emmy Bruns, and my whole lab. And then these are past lab members and some collaborators here. Here. Yeah.
I'm wondering, you had shown us in the beginning that array of all those pretty endophytes.
Yeah, yeah.
How much you think the results you get depend on which of those you choose?
I think it will depend on it, and that's part of this complexity that I want to test. I started out with Fusarium verticilloides because it's the most common endophyte in maize. So if I didn't see an effect with that, then I didn't, I wasn't really sure how much it was worth pursuing further. Yeah. Yeah.
So if I understand correctly, it looks like you're talking about, at least at the plant surface, there's direct competition.
Yeah, yeah.
What if you inoculated on one leaf and did the other one on the other leaf? Would you reduce some of that noise, or what if you put the Fusarium in first one day and
yeah, those are interesting experiments. So we have done, well, first of all, they both basically form local infections. So a long distance on different parts of the plant probably would have to be mediated by plant immunity, and I'm not sure that's transmissible. I'm pretty sure that's not transmissible in this system.
well, if your resistance gene, though, you said went up, that should be transferred.
Well, it's not in all plants. Most plants turn on that resistance factor, and they trigger a reactive oxygen response, and they kill the local cell, which restricts the growth of the pathogen. So it's usually a local effect. But the other question is interesting. I mean, they're both interesting, but the second one, we've actually done some experiments that way, and if you add Fusarium first, it doesn't actually establish in the plant and limit the growth of Eustologo any more than at the same time. If you establish them at the same, if you inoculate them at the same time, you get the largest protective effect. The thing that was, again, counterintuitive was if you inoculate Eustologo and then Fusarium, for whatever reason, you actually get increase in, it almost facilitates it, so you get an increase in infection rate. Yeah, it's small but noticeable. Yeah, so the timing certainly matters. Yeah, yeah, absolutely. Yeah, Tim.
Eustologo is sort of considered one of these classic obligate biotrophs, and so I'm
Yeah.
wondering whether the plant pathology community accepts this idea that it's growing in the soil, or what is your sort of idea of how this competition is going on?
Well, I have isolated Eustologo from the soil. yeah, and Fusarium. So they both occur there. Yeah.
But we know Eustelago probably goes into the soil and maybe just sits there, perhaps.
Right. But I isolate Eustelago yeasts from the cell, from the soil. That it has a haploid phase that's yeast-like, and that I can find and I can isolate that from soil.
So then you think that the Fusarium and the Eustelago are competing with each other?
Yeah, I do. Yeah. Yeah.
Is this information applicable to the agriculture community that raises corn?
Yeah. We did some, so I was curious, like under some models, say if you had all of your maize plants with Fusarium and used that as a biocontrol agent, so you had like a very high percentage of your plants that were had this endophyte in there, it would on the immediate, so then it should reduce pathogen infection and reproduction, right? So to me, that then should select for greater virulence. But with the variation we see in this model, so that might happen. But with the variation we see in our results, you would only get a selection for increased virulence of the pathogen if the endophyte was present in somewhere around 80% of the plants, so it has to be there at a very high frequency. So you might be able to use it as a biocontrol agent, or any of these endophytes as biocontrol agents, and they are being explored for that. But think about what you're going to do then. You're going to put them in all the plants, and you're going to basically engender the same kind of competitive selection for virulence as you do in if you just have plant resistance. You're just substituting endophyte resistance for plant resistance. So I don't actually think this is a great idea.
They haven't come up with a hydrogen corn to fight off this goal?
Yeah, field corn is actually pretty resistant, but the sweet corn we like to eat is pretty susceptible. Yeah.
If there's no more questions, let's thank Georgiana again.
Thank you.